Kai, you're muted. You're muted.
Oh, I see. Okay. Hello, I'd like to welcome all of you to this year's Swirling Lecture, which is an timely and very important topic, the mechanisms and control of COVID-19. I'm co-hosting this year's Swirling Lecture with my distinguished colleague, Dr. Alan Engelman. I would particularly like to thank our three outstanding speakers today, Dr. Rolf Baric from the University of North Carolina at Chapel Hill, Dr. Dan Baruch from Biodiegnes Medical Center, and Dr. Michael Minna from the Harvard School of Public Health. I would first like to introduce the Swirling Lecture. The Susan Swirling Visiting Scholars Fund was established by Phyllis Fifi Swirling Kellam in 1981 in memory of her daughter Susan, who succumbed to ovarian cancer in 1975 at the age of 20. In Susan's memory and to promote Dana-Farber's life-saving work, gifts were donated to support an annual lecture where leading cancer researchers and scholars are invited to share the latest findings with peers. Fifi is an honorary trustee of Dana-Farber, a dedicated member of the Dana-Farber Society, and has volunteered at the front desk of Dana-Farber for over 40 years, serving with great energy and enthusiasm. Every year, she attends this seminar with excitement and earnest hope that the research presented will save somebody else's child. I would now like to introduce our first speaker, Dr. Rolf Baric. Dr. Baric studies a very important topic, the molecular mechanism of viral evolution and virus-host interactions, in particular for coronaviruses and a number of other viruses. He received his PhD from North Carolina State University, did a postdoc at UCLA, and is now the Kennan Distinguished Professor in the Department of Epidemiology, as well as Professor of Microbiology and Immunology. He has spent the last 3 decades as a world leader in the study of coronaviruses, and importantly, alerted the scientific community to threats from emerging coronaviruses. More generally, he has led the world in recognizing the importance of zoonotic viruses as a potential source for new emerging pathogens in humans, with detailed studies of the molecular and evolutionary mechanisms that regulate the establishment and dissemination of such viruses within a newly adopted host. Based on his leadership in this area, he has served on the National Academy of Sciences workshop on emerging infectious diseases for a number of years. Most recently, he's made a number of important contributions to dealing with the current pandemic, including the development of a mouse-adapted coronavirus that has enabled him to perform important preclinical testing of a number of important drugs, including small molecule antiviral drugs such as remdesivir, which is now FDA approved, a number of vaccines, fully human antibodies, and pegylated interferon alpha. So he will now give a seminar which is entitled Preparing for Current and Future Coronavirus Zoonosis Events. Please join me in welcoming Dr. Baric.
Well, it's a pleasure to be here. I thank you all for the invitation. And Phyllis, I thank you for your support for science all these years. You've made a difference, and scientific community certainly appreciates your efforts and support. Again, thanks for inviting me. I'm going to start off with a relatively morbid slide. I apologize for that, but this is a sort of a summary that captures the impact of emerging infectious diseases on human health over several centuries. And in this case, it's showing the death toll to many different viral and bacterial pathogens that have occurred in human populations. And down here at the far left right-hand corner are the emerging coronaviruses, SARS, MERS, and then the COVID-19 virus, which emerged in December of 2019. Subsequently, in about 16 months, has caused 141 million infections globally, resulting in about 3 million deaths, placing it on par with the 17th century Great Plagues. Currently in the US, we have about 570,000 deaths. It's somewhat difficult to predict where this will end. I think the best case scenario may be that the US experiences another 50,000 deaths or so before vaccines and public health measures get this threat under control. That, of course, is mitigated in some ways by the emergence of variants, which may be capable of infecting those with low immunity, and from public distrust of vaccines and vaccination, which may result in a quarter of the population not being vaccinated. Where this epidemic will end, we don't know, but it's certainly running rampant in places like India and Brazil, South America, Central America, and elsewhere in the world. So the outline of today's talk is going to discuss emerging coronaviruses. I'll talk a little bit about variants. I'll briefly discuss our animal models and then talk about our role in the development of therapeutics and vaccines. And obviously here on the right side of the slide, these are coronaviruses in the by electron microscopy. Many of them hang out in bats in general, among the 1,300 bat species on the planet. What little surveillance we have done indicates that anywhere between 6 and 10 unique coronaviruses strains are circulating in a bat species at any given time. In humans, these viruses replicate in the airway epithelium. In this case, these are bronchial airway epithelial cells that have been infected with SARS coronavirus 2. That's the little red dots are the virus, yellow is the mucus, and these blue appendages here are the cilia that line the airways. And this is a diagram of the coronavirus particle. It contains an RNA molecule, positive polarity of about 30,000 nucleotides in length and surrounded by a lipid envelope. Much of the talk will focus on neutralizing antibodies that target the spike glycoprotein and the role of the spike glycoprotein in mitigating cross-species transmission. Now, historically, the contemporary human coronaviruses, of which 3 are listed here, NL63, 2290, and OC43, all are believed to have zoonotic origins, emerging into human populations by phylogenetic studies anywhere from about 120 years for OC43 coming from cattle to about 800 years ago with NL63 from bats. Some believe, based on the clinical symptoms of patients during the Russian flu of 1890, that may have actually been OC43's emergence events, although that's wild speculation. In the 20th century, we mostly saw animal pandemic viruses emerge, including viruses of swine and cattle, like respiratory bovine coronavirus and porcine epidemic diarrhea virus, which arrived in the US in 2012, causing about 10 million deaths in piglets. And then in the 21st century, starting around 2002, we had SARS coronavirus emerge in Southeast Asia from bats, MERS coronavirus in the Middle East from bats, and SARS coronavirus 2 about 16 months ago with the closest relatives, again, in bats. In the interim, we've had 3 emerging swine coronaviruses of the 21st century, including SADS coronavirus, which I'll talk about briefly here in a minute. So there is some acceleration in cross-species transmission movement occurring in this century, and it does suggest that we need to keep our guard up. In addition, there are reports of feline coronaviruses, canine coronaviruses, feline canine recombinant coronaviruses, and porcine delta coronavirus, all being able to cause pneumonia in children, sometimes with some frequency. And so many animal coronaviruses are actually poised to infect human populations and cause disease. Here's one example of this. In 2016, China began to report outbreaks of high mortality respiratory and enteric disease in newborn piglets with about a 99% mortality. They called this virus severe acute diarrhea virus, or SADS coronavirus. When they sequenced the virus, it was a group 1B coronavirus, In other words, it's closely related to two human coronaviruses, NL63 and 229E, which are shown right here. And although the genome backbone is 1B and very close to all these other classic 1B coronaviruses, the spike glycoprotein is quite different. And in fact, it's closely related to a bat coronavirus called HKU2. And so it's thought that this virus emerged from a bat coronavirus and spread into pig populations. We became interested in this virus because there's about 1 billion pigs on the planet, and pigs could be incredibly important reservoir hosts for the emergence and spread of this virus into humans. And so we synthetically resurrected the virus in our laboratory, inserted a red fluorescent protein gene so that infected cells would turn red, like you can see here, and began to ask some fundamental questions. First, we asked whether contemporary human sera from other 1B human coronaviruses, could it neutralize SADs? And the answer is no. We then next asked whether it could replicate in primary cells from the human lung. These are human fibroblast cells, again, can be infected with SADs. Microvascular endothelial cells are susceptible. Nasal airway epithelial cells and bronchial airway epithelial cells can be infected with SADs, as well as primary intestine cells from humans can be infected. Some replicating to high titers of 10 to the 6th, 10 to the 5th in intestine, or 10 to the 6th in fibroblast cells. So these viruses certainly are poised for cross-species movement. And one of the, I think, good outcomes from this particular study is that before this event, the Chinese were studying SADs under BSL-2 conditions, and they've moved all those studies into high containment facilities. There's some commonalities that are associated with coronavirus cross-species transmission. First, they seem to traffic along receptor orthologs. In other words, the same protein in humans, bats, and other species can function as receptors for virus docking and entry. In addition, protease orthologs can also be used as viruses traffic across species. And particularly for the group 2C MERS-like viruses, these proteases are key regulators of cross-species transmission events, as well as the receptor. So in the case of the group 1 coronaviruses, for example, they use CD14 in humans, felines, canines, and other species. Group 2 SARS-like viruses use ACE2, angiotensin 1 converting enzyme 2, and the MERS-like viruses use DPP4. The other drivers of cross-species transmission are the regulated fidelity. The mutation rate of the virus is around 10 to the -6. Although we think when under certain conditions of stress or innate immune suppression, the mutation rate can change. So it's regulated fidelity, and they can evolve much quicker if they need to. In addition, they undergo high-frequency RNA recombination. We showed this back in the late 80s, that during a mixed infection, up to 25% of the progeny can be recombinants that contain genome segments from each of the parental viruses. Now, importantly, the spike glycoprotein is very plastic. It handles deletions, insertions, and mutations. The actual coronavirus world record for a deletion in the spike glycoprotein was set by Michael Buchmeier when he described 150 amino acid deletion in the spike of mouse hepatitis virus that converted it from an encephalitic to a demyelinating virus. And it also completely changed the antigenicity of the virus. So I would say keep an eye out on deletions as these SARS-2 variants emerge over time. Now let's talk about the data that suggested around between 2012 and 2015, that bats harbored a large number of SARS-like viruses that were high threat viruses, or what we would consider high threat viruses. During the 2003 SARS epidemic, the virus was thought to have originated from bats. It was transmitted to an intermediate host, which are civets and raccoon dogs, and in the open market were transmitted to humans, and then human-to-human transmission began to evolve. With the discovery of a virus called WIV16, which is 98% identical to SARS coronavirus 2 in 2015, that suggested an alternative possibility in that the virus could have been transmitted directly to humans, then to civets, and then that set up a transmission cycle to help maintain the virus in human populations. So after the SARS epidemic, in essence, was declared extinct in 2015, we became very interested in asking what sort of SARS-like viruses exist in the bat reservoir, and are any of these threats high threats to human health? Now, the approach that we did this was basically we functioned as, I like to call it, metagenomic pirates, sailing on the sequencing databases of GenBank. And so we would survey sequences and then synthetically resurrect spike protein genes from different members of the group 2B SARS-like coronaviruses shown here. Most of these are all been defined in bats. And then we would synthesize those genes and insert them into a molecular clone, recover the virus, and then we get to ask questions about whether those spike glycoproteins could program infections of primary human cells. And so, for example, these first three, WIV16, WIV1, and SHC014, which are about 90 to 97% identical to the 2003 SARS coronavirus epidemic strain. These spikes could program infection on primary human airway epithelial cells. The viruses could grow as well as SARS coronavirus. They could use the human bat CIVID ACE2 receptor just as well as SARS coronavirus. Although dropping these spikes attenuated the virus in mice, they allowed us to then synthesize full-length genomes and recover full-length viruses. Now, as you move to the more antigenically distinct strains, they begin to escape therapeutics against SARS coronavirus and a blunt vaccine efficacy. So what that data had suggested was that the SARS-like coronaviruses in bats consist of this heterogeneous swarm of related strains that could vary by as much as 35%. Some of these are pre-programmed as generalists to immediately spill into human populations, use human receptors, and replicate efficiently. So we predicted in 2015, 2016 that we would eventually see additional SARS coronavirus related outbreaks in humans, and that they could vary by up to 35%. In 2019, the SARS coronavirus 2 virus emerged, which is 22% different than SARS, which then caused a global pandemic with the epicenter in and around Wuhan, China. Now, both SARS and MERS coronavirus fall in the same category. So you can create these, in essence, libraries or encyclopedias of high-risk sarcocoronaviruses that can emerge and spill over into human populations, including the SARS coronavirus 2003 strain here, which is 35% different than these clade 2 bat coronavirus strains called HKU3. The other clade 1 SARS-like strains that we've identified are WIV1, WIV16, and SHC014. That's just not 3 viruses. Each of those represent a swarm of related viruses that range anywhere from 98% identical to SARS to about 88% identical to SARS. SARS coronavirus 2 is about 22%. They all have the same features. They can grow in human cells. They can use human ACE2 for entry. They cause ARDS in animal models. They cause an age-related disease gradient. As you get older, you're more vulnerable. And as you increase the amount of variation in the spike, you begin to escape existing immune therapeutics. An important take-home message here from studies on growth on primary human cells is that mutation-driven evolution may not actually be required for colonization of a new species. The triggering event may simply be a human being coming into the context of bats that have the virus, and then that human being in a position to be in a dense population to allow for emergence and spread. The MERS coronaviruses are very similar. There have been 2 that have been identified that can replicate efficiently on human cells. This is MERS coronavirus here, and a MERS-like bat coronavirus that we're still about to write up and finish writing up and submitting. That's 35% different than MERS coronavirus. We have made a molecular clone and put in a green fluorescent protein. And you can see it grows quite well on human airway epithelial cells. It uses the human DPP4 receptor. And it's quite different. Antisera against MERS does not neutralize effectively this highly divergent bat strain. So another reason why the threat risk is high is when you examine the phylogenetic tree of coronaviruses. So if you look at the 2A coronaviruses, there are two contemporary coronaviruses that co-circulate in human populations, OC43 and HKU1. There's two contemporary group 1B coronaviruses that circulate, NL63 and 229E. So they don't provide cross protection in either case. And that suggests that there's plenty of antigenic space in the 2C and the 2B coronaviruses for two or more strains to emerge from animal populations and colonize the human host. And so universal vaccine design, I think, is critical. Now, originally, the SARS-2 virus that emerged out of Wuhan was pretty much a single lineage. In February of 2020, a variety of individuals who were tracking genome variation that occurred over time began to identify the emergence of a new variant called G614, which is shown here. It was first identified in Southern Europe, but it was seen elsewhere as well. And by late May of 2020, it had become the dominant world globally. Betty Korber showed that this G614 mutation was associated with higher respiratory tract viral loads in humans, but didn't seem to be associated with increased severity. So we took advantage of the reverse genetics molecular clone that we'd made in the lab to begin to ask questions about the role of that mutation. I'm just going to show a couple of figures because it's been published. First, we made isogenic viruses with and without nanoluciferase that encoded the G variation. We then asked whether those wild-type viruses, how well they grew in nasal and bronchial airway epithelial cells. And I'm just showing the nasal airway epithelial cells, and you can see here that you saw significant increases in the G variant growth in the nose, or cells from the nasal airway epithelium, at 24, 48, and 72 hours. If you do competition experiments, head-to-head competition experiments, the G variant always outcompetes the D variant, explaining its capacity to dominate globally over time. We worked with Yoshiko Oka to ask questions about whether the G mutation affected transmission, airborne transmission. Now, in this particular experiment, Yoshi's got animals, hamsters, donor hamsters that are infected with virus. There's an airspace so that airborne transmission is the only route that can occur by which this recipient animal can be infected. Now, if you infect 8 donors, either with the D variant or the G variant, shown here in gray and black, they replicate in the donor animals at about the same titer and clear about the same rate. If you look at transmission to the exposed animals, the G variant is transmitted significantly more efficiently by day 2 as compared to the D animals, although eventually the D variant infects all the hamsters. So our data suggested a variety of things. It suggested the virus was about 2-fold more sensitive to neutralization as compared to the original Wuhan strain. Others showed that as well. It's more transmissible. It replicates better in the nose, where it would be in a good position to transmit. And others have subsequently gone on to show that that G mutation really does have a tendency to open up the RBD structure so it can grab onto the ACE2 receptor. Now, this G variant was the progenitor to all the current variants that are circulating. The list is quite long. I'm only going to put a few up here, including the UK, the South African, and the B1 Brazilian strain. All of those are associated with increased transmission. The UK strain has the best data for increased virulence. It's likely that the South African and the B1 strain are also more virulent. Although most of these mutational changes don't have a big effect on neutralization, on the reduction or the change in the neutralization efficiency, the South African strain is much more resistant, 6 to 10-fold times more resistant than the other strains. This is just shown here with some data from our lab, where we've have a B1351 variant. The spike's been dropped into an nanoluciferase encoding recombinant virus. And we see a 4 to 6-fold reduction in neutralization titers using this, in this particular cases with mRNA vaccinated mice, but we have similar data with humans. And with strains isolated at UNC, they grow equally well, either UK or South African strains, in both bronchial and nasal airway epithelial cells. Now, we've, using these vaccinated mice, which are shown here, just focus on the gray and the red on this bottom part, and I'll talk about the rest of the slides later. We built this 351 variant in our mouse-adapted backbone that has changes elsewhere in the genome, but not in spike. This virus causes about a 15 to 20% drop in weight loss, depending on the age of the animal by day 4 or 5 post-infection. But if you vaccinate with a replica of the Moderna mRNA vaccine, you get complete protection. You also get complete protection from virus replication in the lung, and also complete virus replication from virus replication in the nose. So our data argues that the existing Moderna vaccine provides relatively robust protection against the South African variant, at least in the mouse models of human disease. Now I'm going to go to discuss about therapeutics. Our lab was involved in developing remdesivir, EIDD2801, and pegylated interferon lambda. Two of these, remdesivir and EIDD28, are pan-coronavirus drugs. We've also were involved in the development of Eli Lilly's therapeutic monoclonal antibody, the antibody from Jim Crow's lab that has been marketed by AstraZeneca, and we worked with Lara Walker to develop ADG2, which is in phase 2 trials. This is a pan-SARS-like coronavirus antibody that is in clinical trials. Our mouse model is based on the following data. The original Wuhan strain cannot replicate in mice. We mouse adapted it by engineering in mutations that would allow it to bind to the mouse ACE2 receptor. That replicated and caused mild disease, but with passage in mice in vivo, you ended up with a lethal model. And to summarize what we think is the mechanism of high disease and mortality following SARS coronavirus 2 MA10 infection, we're going to focus on virus infection in the alveoli. LAMP3 is an AT2 marker. Red is a viral nucleocapsid marker. You can see co-localized signals, so AT2 cells are the primary target. It's quite striking what happens in the animal when you look across the entire section. In this case, the white is AT1 cells, the green are AT2 cells. Red is an infected animal now. And notice that all the green signal just disappears. This is surfactant C that has just completely been ablated from virus-infected cells, as well as surfactant B. This doesn't begin to become restored in the lung till about day 7, and is still restoring at day 15 to 30 post-infection in survivors. And so we think mortality here is associated with virus infection of ciliated airway epithelial cells, club cells in the terminal bronchi and the AT2 cells. We see acute lung injury in ARDS in these animals. There's a cytokine storm, and we think surfactant deficiency is playing a major role in early death in the animals. Now, I want to talk about, this is a complex study, which I'm going to use it to illustrate a point. We actually did head-to-head comparisons with remdesivir, human monoclonal antibodies, and then studied them in combination. And so basically our approach is to infect a mouse with either treated with vehicle, either prophylactically with one of these agents, or then therapeutically up to about 48 hours post-infection, and then study a variety of metrics. And you get near identical outcomes with therapeutic antibodies. So I'm just going to show remdesivir here. If you give remdesivir prophylactically, notice that the animals don't lose any weight. In contrast, if you put vehicle in gray, they lose 20% of their body weight by day 5, and many animals have begun to die. Therapeutically, remdesivir works great at 12. It's still effective at 24 hours, but it fails by 48 hours, shown here in blue. That's not only affecting weight loss, but you see the same thing with virus titer in the lung. And this is it's a lung hemorrhage score, but it's really a lung discoloration score that shows that, again, any direct acting antiviral that we give has to be given very early for it to be effective. And similar findings are being identified and have been subsequently shown in humans. I should just point out that we noticed this back in 2017. That early administration of direct-acting antivirals would be critically important for control of disease. Why is that? Well, in an individual infected before symptomatic disease at time 0 here, About 4 days before they've been infected, you have very high virus titers early in infection, and then they begin to fall off. We and no one else, I think, has been able to isolate virus after day 10 post-infection, unless the individual is immunosuppressed. If you look at the severe disease phenotype for ARDS, it occurs after acute virus infection. So you have a biphasic early high titer replication, late disease manifestation. So remdesivir, which is given by IV drug use, is given late in the therapeutic window. And so it has a modest effect in terms of improving time to recovery. What's needed are therapeutics that drive this, the effectiveness of the drug further into the therapeutic window. One way to do that is with oral therapeutics like EIDD-2801 or molnupiravir, which is an RNA mutagen. It's given as an oral tablet. It has the same phenotype in mice in terms of giving orally. It's effective if given up to about 24 to 36 hours post-infection. Here's the vehicle control, here's prophylactic, and then the different therapeutic windows. Again, by 48 hours, it's not effective. The beauty of remdesivir is at the time of symptomatic disease and diagnosis, you can get that given as an oral tablet in a outpatient setting. Working with Ridgeback Biosciences and Billy Fisher, who's the lead investigator on this phase 2 outpatient study at UNC. This is data that was published, that was presented by Ridgeback at CROI. This is a summary of that data where patients, again, at diagnosis prior to serologic conversion were given oral EIDD-2801. By day 3, there was a reduction in the number of individuals who still had live virus that could be cultured from those individuals. By day 5 post-infection, all of those on remdesivir had cleared infection. And this is just the individual data where patients given 200, 400, or 800 milligrams of drug. Now, because of this data, Ridgeback, who sold the drug to Merck, have halted the phase 2 study, and they're moving this drug into phase 3 studies. And hopefully we will have an oral version of this, as well as an oral version of remdesivir sometime shortly to be given in an outpatient setting. The therapeutic antibodies that we work with Laura Walker are shown here in red. Again, this was these were achieved by directed evolution of antibodies that had some broad neutralizing activity against multiple sarcocoronaviruses. Notice that this was effective against SARS coronavirus 2. These other antibodies include things like the Lilly antibody and the Regeneron antibodies. As you move into SARS coronavirus, many of these antibodies have no activity. The 309 active antibody loses about 100-fold activity. And as you move into the most distant sarcocoronavirus from SARS-2, all these other antibodies really fade away in terms of effectiveness, while ADG2 is still effective. And this is just some prophylactic data showing that the antibody protects against both the SARS coronavirus 2003 strain, shown here in red, and the MERS, the SARS-2 MA10 virus, shown here in the lower panel. This is respiratory function, again, protecting against loss of respiratory function, reducing clinical congestion scores, and completely protecting against virus replication. Now, in terms of vaccines, we've interfaced with a variety of groups, including Moderna, Janssen, NIH, and Peter Palese with his NDV vaccines. I'll talk a little bit about our chimeric spike vaccines, but I just wanted to show, I think, 1 or 2 key slides to finish up. Again, I'm reminding you here that the viruses that I'm going to talk about are Bonnie's SARS coronavirus here, which is 22% different than the SARS-2 strain shown here. And these 2 bat strains are about 8 to 10% different from SARS and over 25% different from SARS coronavirus 2. Now, one of the questions that we had to answer early for getting the Moderna vaccine into phase 3 trials was to ask the question whether the mRNA vaccines could elicit a Th2 immune pathology that had been described previously by our group with the NIH doubly inactivated vaccine that had been developed against SARS coronavirus 2. So working with Sarah in our lab, working with Kizzy Corbett and others at the VRC, vaccinated mice with the mRNA Moderna vaccine at 0.1 or 1 micrograms of mRNA, or the doubly inactivated SARS coronavirus vaccine. When you infect these animals, notice that at 0.1 and 1 micrograms, the Moderna vaccine protects against SARS coronavirus 2 M810 challenge, but the doubly inactivated vaccine against SARS coronavirus did not protect at all. It also did not protect against virus replication, while the Moderna vaccines that were against the homologous SARS-2 strains were completely protective. Now, importantly, the doubly inactivated vaccine resulted in a Th2 immune pathology following SARS coronavirus 2 M810 challenge. We saw increased cytokines, which are shown across the bottom of the panel, including IL-4, IL-5, IL-13, the ataxin. There were large numbers of eosinophils that had moved into the lung, and there was evidence of Th2 immune pathology only in the doubly inactivated vaccine infected recipients, not in the Moderna vaccine recipients. So this Moderna vaccine did not drive any kind of Th2 immune pathology. What about its effectiveness against different sarcoronavirus strains? Well, in this case, vaccinating mice with the SARS coronavirus 2, a replica of the Moderna vaccine gives a ID50 of about 1 to 10,000 against SARS coronavirus 2. There's a 40-fold reduction against SARS coronavirus, about a 300 to 500-fold reduction against the SHC014 strain, and about a 50-fold reduction against the WIV strain. So the effectiveness here against other sarcoronavirus strains is extraordinarily low. You can ask the question whether SARS coronavirus vaccine, 2 vaccines, will protect against these other clade 1 sarcoronaviruses like SARS or SHC014 or WIV1 infection, or other clade 3 strains or other clade 2 strains. And so I'm going to talk about 2 vaccines. This is the homologous monovalent vaccine. But we've also built chimeric spikes that have different component parts from different clade 1 coronaviruses delivered by mRNA-based vaccines. So for example, the NTD domains from clade 2 HKU3, the RBD domains from the clade 1 SARS strain, and the rest of the molecule from clade 3 SARS coronavirus 2 strains. You could also prime or prime boost with a heterologous spike that is mostly SARS-2, but has the SHC014 receptor binding domain. And the idea here is by mixing and matching, you can focus responses to specific domains, and you can expand the breadth of the response with a minimal number of component pieces. Here's another example, again, where you have 2 heterologous components where you are hoping to drive or focus the immune response on the conserved regions between different prime boost regimens. Now, just to show some of this data quickly, in the case of a SARS, you can vaccinate with different formulations. I'm not going to go into the detail. This is a mock vaccinated animal with a Norwalk capsid protein. It does not protect. In this case, the homologous SARS-2 vaccine protects against SARS-2 challenge, as does various combinations of these chimeric spike vaccines. They protect against weight loss, titer, a little bit of breakthrough in the nose in terms of the nasal turbinates. You can also then challenge with SARS coronavirus from clade 1. In this case, again, the control animals lose over 20% of their body weight. In this case, in red is the SARS coronavirus 2 homologous vaccine. It does, like the doubly inactivated vaccine, it does not protect against SARS coronavirus 2003 challenge. Interestingly, one of the group 3 vaccines here shown right here, which has SARS coronavirus 2 in most of the component of it, and then a clade 1 SHC014 spike, does provide some limited protection from weight loss, but not replication either in the lung or in the nasal turbinates. And neither does the SARS coronavirus 2 vaccine provide any protection in the lung or the nasal turbinates following challenge. Only these other universal combination vaccines provide complete protection. Now you can also ask about heterologous challenge. In this case, we can now challenge with the 351 variant. Again, here's the mock vaccinated animals. The SARS-2 vaccine provides complete protection, like I said before, as do our other chimeras. Notice that this group 3 component, which is mostly SARS coronavirus 2 in the NTD and the S2 region, but has this very, very different RBD domain from clade 1 SHC014, provides some partial protection, not only from weight loss, but about a 1.5 log reduction in titer in the lung and protection in the nasal turbinates. So you do get some cross protection with this particular chimera, suggesting that NTD-ST domains are eliciting antibodies in this chimera that can protect against the variant. A WIV challenge could be another alternative model. And again, the full chimeras completely protect. This bivalent group 3 provides partial protection from replication, but the SARS-2 vaccine does not. So in summary, I think universal SARS coronavirus vaccines are possible. The current messenger RNA vaccines protect against some variants of concern in mice, and that in this particular case, immune responses probably targeting NTD and S2 are driving that protective response that partially attenuates pathogenesis. So with that, I thank you for your attention. I know I ran over a little bit. We work with a large number of different groups, and I'll be glad to take some questions.
Great, that was very, very interesting, very insightful. So how do you, what I found interesting is that you described the bad coronavirus not as distinct strains, but as a swarm. So given that great genetic diversity, what is the most effective approach of developing broad vaccines? I know you presented some ideas on that, but do you think that repeated vaccination will be necessary to deal with new emergent strains?
I think one of the fundamental questions, which I wasn't completely willing to talk about and show about the data today, was that the SARS coronavirus 2 mRNA vaccine does not protect against SHC014, WIV1, or SARS coronavirus challenge. There is evidence of eosinophilia and Th2 skewing of the immune response. And so I think we desperately need to develop universal vaccines because of that. So how do you develop that? Well, one of the whole reasons we became interested in bat coronaviruses was that if you're going to develop universal drugs or vaccines against a family of viruses, you needed a repertoire of viruses to demonstrate efficacy, at least both in terms of being able to grow in primary human cells, but also being able to grow in mice. And many of the strains that I just talked about also replicate in primates. And so those reagents, in particular, specific strains in clade 1, clade 2, and clade 3 of the SARS coronaviruses provide a relatively robust portfolio of strains that are highly heterogeneous and that'll give you an idea how well they work. Now, will there be potential variants that can break through? Possibly. Yeah. So we, for example, are always on the lookout for SARS coronavirus strains in particular that have 35% variation. We think there are strains out there in bats that are 35% different, and that we desperately need one of those viruses for evaluating vaccine and drug performance and therapeutic antibodies. So
great. And have you tried in the mRNA vaccine format to combine sequences from highly divergent coronaviruses into a single vaccine, or is the antibody response against each of the distinct antigens attenuated when you develop such a combinatorial vaccine cocktail?
Yeah, there's a couple of different approaches for universal vaccines. One would be different receptor binding domains that could be used in various prime boost regimens, or built on the easiest way for me to say it is linked on a string together. And if you can solve the immunodominant problems there, I think you'll have a good vaccine, at least in terms of antibody responses. Another approach is to identify universal antibodies with breadth that target specific epitopes that you can focus immune responses on using structure-guided engineering, which I think is a good approach. We're interested in just the whole plasticity of the spike glycoprotein, which is one of the reasons why we're interested in building chimeric spikes with different component parts. And there are 3 that you can play with at a minimum. You can probably break that down into more based on further division within a specific domain like the RBD or the NTD. And most people argue that the RBD is the most important domain. I'm less certain of that. I think the NTD is a critical domain with that generates highly potent neutralizing antibodies. I think to some extent that the lentivirus pseudotype systems are underrepresenting their potency, which is why I like live virus neutralization assays. I could be attacked on that, and I will be somewhat vulnerable. I accept that statement. But the other thing, I think, the other reason why I believe the NTD is really important is there's so much evolution occurring in NTD. That doesn't occur unless there's immune selection there, and I think it's important immune selection. So I believe that you could build a couple of different spikes to try to minimize the number of component parts that would drive sort of a global response against the spike glycoprotein, with focusing it on specific regions, depending on how you design it. So there's a lot of different approaches. Overall, I'm encouraged in terms of our ability to make a broadly effective SARS coronavirus vaccine. Universal coronavirus is a whole nother question.
Okay, great.
Ralph, maybe one final quick question. Of course, vaccine is always the best if it can work, because you can keep people from getting sick in the first place. And with today's pandemic, vaccines have been really highly focused. But you also brought it up in terms of, let's say, a universal small molecule inhibitors, for example, of critical enzyme active sites that might be representative across the virus family. What do you think about that in terms of universal small molecules?
Both remdesivir and molnupiravir work against all the SARS strains we have in our portfolio, all the MERS strains, representative group 1 strains, and even the group 3 coronaviruses, the porcine delta coronavirus. So those have, again, the broadest portfolio of viruses that are highly heterogeneous and different. Those drugs work against, and that's why I'm so hopeful that an oral drug will be available. Treatment of acute infection is certainly an important issue, but I think at least for the United States, after vaccination, the real major concern is going to be the chronic disease phenotypes that are occurring in patients, the chronic kidney outcomes, cardiac. Our own group is heavily focused on chronic respiratory disease like pulmonary fibrosis and airway disease. These are all types of, and neurologic disease. These are all types of hit and run diseases that could continue to kill people for the next 10 years. And if something like pulmonary fibrosis, which has all the same risk factors for COVID-19 infection and idiopathic pulmonary fibrosis, and many common susceptibility loci in humans, we could be on the verge of a pulmonary fibrosis disease wave 3 to 5 years from now, which could kill 5 to 10% of the survivors. That would be a horrifying outcome. So I think one of the things the scientific community desperately needs to do is develop model systems for these chronic diseases so that we can begin to test drugs. I mean, the therapies for pulmonary fibrosis run $100,000. That's going to be a little tough for the healthcare system to handle.
Thank you very much, Dr. Barack. I think we have to move on to the next speaker. So it's my great pleasure to introduce our next speaker, who is Dr. Dan Barouch. Dr. Barouch is the William Bosworth Castle Professor of Medicine and Professor of Immunology at Harvard Medical School. Dr. Barouch is also the director of the Center for Virology and Vaccine Research at the Beth Israel Deaconess Medical Center. Dan is a worldwide leader in the design and employment of effective vaccine strategies against emergent viral diseases and worldwide pandemics. His lab in particular has leveraged viral vaccine strategies based on adenovirus serotype 26, which have advanced to phase 3 clinical trials for HIV/AIDS and are currently used to prevent COVID-19 disease. We therefore are extremely fortunate to have Dan here today to present his talk on COVID-19, developing a vaccine during a pandemic. Dan, please.
Thank you very much, Alan. Can you hear me and can you see my first slide?
I can. I think
Very good. So what I'd like to do in the next 30, 35 minutes is to really give you a bird's eye perspective of sort of a retrospective of, I guess, our personal journey over the last year, helping to develop one of the 3 vaccines that currently has FDA authorization. And I'll make some comments about the vaccine field as a whole, and then I'll also describe what we've done over the last year in helping to develop the J&J vaccine. And I'll make some comments at the end about the recent widely publicized news about thrombosis and where that might be going. So as people know, a new disease emerged in December in 2019 that was initially a pneumonia of unknown cause in Wuhan, China. And then very quickly, it was determined that this was a novel coronavirus, which was eventually called SARS-CoV-2. On January 10th of last year, the sequence of SARS-CoV-2 was released by the Chinese researchers, and we and many other groups decided very quickly to start vaccine development because of the potential this virus appeared to have to cause a pandemic. At that point in time, there were 41 cases of SARS-CoV-2 reported and 1 death, and we consider those numbers to be alarming. Just think about it, just about 14 months ago, 41 cases and 1 death was alarming enough to set off multiple concurrent vaccine development programs. Now, instead of 41 cases, we have 141 million cases. And instead of 1 death, we have over 3 million deaths, with the largest burden of deaths being in the United States. So the scientific community has responded in unprecedented ways. For vaccines in particular, the WHO has been tracking the development of over 200 vaccine candidates worldwide. These essentially reflect all current vaccine platform technologies, including DNA, RNA, proteins, vectors, inactivated viruses, and other platforms as well. But I would argue that the timeline to show clinical efficacy is actually not the most relevant timeline. The most relevant timeline for this pandemic is the timeline to produce and deploy billions of doses of vaccines worldwide, which has not yet been done. In the United States, there have been 3 vaccines approved for emergency use, including the 2 mRNA vaccines, first from Pfizer and then from Moderna, and then more recently, the ad vector vaccine from J&J. We published a review together with our colleagues from Moderna recently on novel vaccine technologies concentrating on RNA and ad vectors. And these are just some figures from that review article. But basically, RNA and ad vectors are very conceptually analogous technologies. They're both called so-called gene-based vaccines because they use only the immunogen of interest rather than the whole virus. A traditional vaccine is either an attenuated version of the organism or an inactivated version of the organism. Here, as you probably know, for RNA vaccines, it's the RNA of the COVID-19 spike protein that's encapsulated in a lipid nanoparticle as the delivery vehicle to be delivered into the vaccine formulation. Ad vectors are conceptually analogous, except instead of RNA, it's the DNA of the COVID-19 spike protein. And instead of a lipid nanoparticle as the carrier, it's an inactivated ad vector capsid. So basically, both of these include nucleic acid of the COVID-19 spike protein with a delivery vehicle. The vaccine world was essentially took a giant leap forward on November 9th of last year, when Pfizer announced the first results from a phase 3 trial showing strong protection of their mRNA vaccine against symptomatic COVID-19. Shortly afterwards was a similar announcement from Moderna with their RNA vaccine. And then at the end of January, J&J announced positive results with the Ad26 vector as well. So in the United States, we have essentially 5 large vaccine programs that at least are or were headed towards efficacy and approval and distribution pending results. The first 2 are the ones I talked about already, the mRNA vaccines from Moderna and Pfizer. These phase 3 trials were launched in July, and the efficacy was reported in November, and FDA granted emergency authorization in December. The J&J Ad26 vaccine was next in line. The phase 3 trial was launched in September, and efficacy was announced in January, and FDA emergency authorization was given at the end of February. The AstraZeneca ChAdOx1 vaccine, the study was launched in August. Because of a number of delays, the efficacy was announced last month in March, and this has not yet received FDA emergency use authorization. And finally, the 5th program is an adjuvanted protein vaccine from Novavax. Phase 3 trial was launched in December, and efficacy is should be announced hopefully, if not this month, then hopefully in May. These are the programs in the United States, and there's another probably half dozen programs from other vaccines around the world as well as these. So vaccine development for a new pathogen typically takes many years or decades. So how is it possible that not one, but multiple COVID-19 vaccines were developed in just about a year? That's truly unprecedented. There's no precedent that even comes close in the history of science globally. I would argue that these vaccines were actually not created in a year. They were actually created over decades, because each of these vaccines reflect decades of progress and advances in virology and immunology, and in particular, the development of gene-based vaccine platforms, particularly RNA and vectors. In addition, these vaccine platforms were already tested for other pathogens, so they could go into clinical trial testing very quickly. And all of the manufacturers performed development activities in parallel rather than in series, with substantial tolerance of programmatic and financial risk, but no compromise in patient safety or regulatory integrity of the clinical trials. And one example of this is the mass production of vaccines while clinical trials were in progress. So the day of FDA approval, there's already millions of doses available to distribute. So our program, in collaboration with J&J, involved the development of an Ad26-based COVID vaccine. The rationale was that we had created replication incompetent Ad26 vectors about 15 years ago, not for coronaviruses, but rather for other pathogens, initially HIV. So the Ad26 HIV vaccine, which is actually still in phase 3 trials, was the original rationale for the development of this platform technology. And we might actually get the results of our phase 2B or phase 3 HIV vaccine trials perhaps this summer. Our first use of this in a pandemic response setting was with Zika in 2016. And with Zika in 2016, we showed that this vaccine platform with a more so-called well-behaved virus actually could raise antibody responses in nearly everyone after a single shot. So this is what gave rise to the concept of the use of a single shot vaccine. J&J then developed an Ad26 Ebola vaccine and demonstrated that mass production and global distribution was feasible without the need for a sub-zero frozen cold chain. They developed formulations that could be stable in a regular refrigerator, so therefore avoiding the need for a frozen cold chain, which we think facilitates distribution to much of the developing world. Ad26 RSV was also developed and administered safely to infants down to 4 months of age. So this is data from our Ad26 Zika study, in which we gave a single shot or a 2-shot version of this vaccine. And the single shot vaccine at 2 different doses are shown in the circles and the squares. And you can see a single shot of this vaccine raised Zika neutralizing antibodies in essentially everybody. And these lasted for at least a year. A second shot of the vaccine given on day 57, shown in the triangles, could boost these responses substantially. So for Ad26 Zika, we learned that a single shot vaccine was a feasible concept, but a 2-shot vaccine would be more potent. So this is what we did. We started working on this project on January 10th of last year, when the Chinese researchers released a sequence of SARS-CoV-2. We actually started working on it that same evening and started to design electronic sequences that could be used as immunogens. So when we got back to the lab on Monday, January 13th, we were in a position to order synthetic genes to help design the vaccine. By the end of January, we had a collaboration agreement signed with J&J, and we started immunizations in small and large animal models in February. By the end of February, we had a virus challenge stock produced, and at the end of May, we had our first results on natural and vaccine immunity in non-human primates, which we thought was important for the global vaccine effort, not specifically this vaccine candidate. For this specific vaccine candidate, phase 1/2 studies were initiated at the end of July, and we also reported the results of Ad26 vaccine protection in non-human primates in July. By September, we started the single-shot phase 3 clinical trial, and 2 months later, we started a second phase 3 trial involving a 2-shot version of this vaccine. On January 29th of last year, interim safety and efficacy data were announced, and at the end of February, FDA granted emergency use authorization. And during the course of 2021, J&J has a commitment to produce and deploy at least 1 billion doses of the vaccine worldwide. So I'll just show a few slides from our paper from July showing that a single shot Ad26 vaccine could protect against SARS-CoV-2 in rhesus macaques. We looked at not one, but actually 7 different versions of this vaccine, encoding different versions of the spike protein. And I'll just concentrate on the bottom one here, which includes a wild-type leader sequence, a full-length spike, and then a furin cleavage site knockout, and the diproline stabilizing motif that's used in 4 out of 5 of the vaccine programs in this country, described originally by McClellan and colleagues. Following challenge, the vaccine that we called in this paper S.PP, which is the vaccine that ultimately went into humans, showed complete protection against viral replication in the lung and bronchoalveolar lavage compared with a large control group that all were infected with SARS-CoV-2 and had viral loads, mean viral loads in the 4.5 to 5 log range. If we look at the other suboptimal vaccines, then these also gave substantial protection with 0 to 2 breakthroughs per group. And the breakthroughs generally were lower in magnitude and shorter in duration than the control group. In the upper airways, which is typically harder to protect in nasal swabs, the vaccine also gave substantial protection. Compare the sham control group, in which all the animals were infected and had high levels of virus for about 10 to 14 days. In the vaccinated animals, the optimally vaccinated animals, most animals were completely protected, and 1 animal showed a low blip of virus for 2 days. The other vaccines also showed partial protection in the upper airways, although perhaps less so than in BAL, with 1 to 3 breakthroughs per group. And the breakthroughs here in the nasal swabs were generally higher in magnitude and longer in duration than in bronchoalveolar lavage. So to summarize the protection, peak viral loads in BAL nasal swabs were about 5 to 6 logs, and the SPP vaccine gave complete protection in the lungs and near complete protection in the upper airways. Because of the variable outcome of the study, with multiple different vaccines being tested, we were able to do a correlates analysis, and we showed that the neutralizing antibody titer induced by the vaccine even 2 weeks after vaccination, and even better at 4 weeks after vaccination, inversely correlated with peak viral loads, here shown in lung, but also shown in nasal swabs. So this study showed that a single shot Ad26 vaccine could protect macaques against SARS-CoV-2, resulting in complete protection in the lung and near complete protection in the upper respiratory tract. We reasoned that a single shot vaccine would have major advantages over a 2-shot vaccine for mass immunization and pandemic control, particularly without the need for a frozen cold chain. We also reasoned that a second shot would likely increase neutralizing antibody titers and therefore might be superior in the end. We also showed that neutralizing antibody titers were the strongest correlate of protection, consistent with a prior study that we did with first-generation DNA vaccines. And so the development plans for this vaccine, which was renamed Ad26-CoV2S, involved phase 1/2 studies that were started at the end of July, which then led into a phase 3 study initiated at the end of September. So these are data from the phase 1/2 studies, 2 papers in the New England Journal and JAMA outlining the immunogenicity and safety of this vaccine. And these are the antibody responses in humans seen after 1 shot and 2 shots of this vaccine. The binding antibodies by ELISA are shown on the top, and the neutralizing antibodies are shown on the bottom. Cohort 1A is younger adults, and cohort 3 is essentially a matched cohort of older adults over age 65. If we just concentrate on cohort 1A for binding antibodies, they're divided and randomized into 5 different groups. Placebo group, groups that received a single shot of the 5 × 10⁻⁻⁻ dose of the vaccine, 2 doses of this vaccine, a single shot of 10 to the 11th viral particles of the vaccine, and 2 shots of this dose of the vaccine. Essentially, everyone developed binding antibodies, or almost everyone developed binding antibodies by day 29. In those that received only a single shot of the vaccine, these titers actually continue to go up from day 29 up through day 71. But in the groups that received the boost, these titers went up by 3 to 4-fold. Similar responses were seen on elderly adults. And the neutralizing antibody responses really parallel these binding antibody responses in that almost everyone developed neutralizing antibodies by day 29, and I think everyone by day 57. But it is noteworthy that the responses, even with a single shot vaccine, continue to go up from day 29 to day 57 to day 71. And we think even further beyond that. With a boost, these titers went up about 3 to 4-fold, and similar titers were seen in elderly individuals. In addition to the antibody responses, vectors are particularly good at generating T-cell responses. So by 2 weeks, and even more, not shown here, by 4 weeks after the vaccination, then the majority of people develop both CD4 T-cell responses, shown on the top, and CD8 T-cell responses, shown on the bottom. We think that CD8 T-cell responses may be particularly relevant for protection against viral variants that partially evade antibodies. So the phase 3 study that was done was called COVE3001. It was a randomized double-blind placebo-controlled trial of this vaccine. It was fully enrolled in December with 44,000 individuals that had a 1-to-1 vaccine placebo randomization that tested a single shot at the dose of 5 × 10⁻⁻⁻ viral particles. And one of the unique things about this trial is that it was truly a global trial. In the United States, in 3 main geographic areas, in the US and South Africa, and in Latin America, including Brazil. When we started this trial, there wasn't any problem of viral variants. I'll also just outline the 2-shot phase 3 trial, which is COVE3009, which is the exact same design in 30,000 individuals, vaccine 1-to-1 vaccine placebo randomization, except testing a 2-shot version of this vaccine. So the same vaccine, but given at week 0 and week 8 in multiple different countries in the US and Europe and South America, South Africa, and the Philippines. So when we started these phase 3 trials, then there really was very little discussion about variants other than the D614G variant that Dr. Barrick described in great detail. But there wasn't any concern about so-called variants of concern. But something extraordinary happened during the course of these phase 3 trials. And not only during the course of these phase 3 trials, but in the exact regions that we were conducting these phase 3 trials, which was the emergence of new SARS-CoV-2 variants that could either enhanced transmission rates, which has been the concern for the B.1.1.7 variant that originated in the UK, as well as could partially evade antibodies, which is the concern for variants such as B.1.351, which originated in South Africa. And these variants emerged essentially during the efficacy collection period of the study. So just to make sure that people are aware, these variants are not only found in faraway lands, but actually now are the dominant virus seen in the United States. This front page New York Times article from a week or two ago is reporting, also, of course, the CDC is confirming this, that more than half of new cases in the US are now due to the B.1.1.7 variant. This is showing increased contagiousness and possible increased pathogenicity. It's less of a concern for vaccines because it doesn't appear to have very many immunologic properties that are different. The variants that we're more concerned about are the variants that contain mutations that also Dr. Barrick described in great detail, mutations in the RBD or the NTD domains, such as the characteristic E484K mutation that can lead to partial evasion of neutralizing antibodies. And these are the virus populations in South Africa. And you can see that up until September, October, it was really quite boring. It was really just sort of a nondescript virus populations. But then in South Africa, the B.1.351 variant emerged and rapidly dominated the virus population, so that during the efficacy collection period of the J&J phase 3 trial, it essentially had emerged out of nowhere and then became the dominant virus. So much so that in the J&J phase 3 trial in South Africa, over 95% of sequenced viruses were B.1.351. Similarly, in Brazil, a similar takeover of the virus population had occurred with both the P1 and P2 variants, which have similar 484 mutation as this B.1.351 variant. And so during the course of this phase 3 trial, it essentially was a direct test of the vaccine in probably the most challenging epidemiology possible, which is in country in South Africa and in Brazil, with all of the comorbidities associated with that, such as a high prevalence of HIV infection. Also, essentially a fully powered phase 3 trial against the P1 and P2 variants in Brazil and against the B.1.351 variant in South Africa. So the results of this phase 3 trial were reported to the FDA on February 26th. And this study was conducted in multiple countries at the peak global surge, the peak winter global surge, including in Brazil that had 70% of the viruses of the P2 lineage, and South Africa with 95% of viruses of B.1.351. Despite this challenging epidemiology, there is still robust protection against any symptomatic infection, 72% in the US, 68% in Latin America, and 64% in South Africa. And actually in the US, there was already a substantial fraction of viruses that were the partially resistant so-called California variant as well. Probably the most important results from this study is that there was robust protection against severe disease of 85%, and that was seen globally. That was seen regardless of geography and regardless of variants, and 100% protection against hospitalization and death in this study, also globally. So this study was one of the first ones that actually had a fully powered phase 3 study against the resistant variants in Brazil and South Africa, and showed robust protection, particularly against severe disease. So these are the actual data from the phase 3 trial. By day 14, there was already substantial efficacy against severe disease of 76%, and by day 28, it had increased to 85%. So this is the Kaplan-Meier curve showing the protection against severe COVID disease in this study. You can see these curves separate very quickly. They separate by day 7. So there's already substantial protection 7 days after a single shot vaccination. And the predetermined time points to look at in the study was 14 and 28 days. So that's what's being reported. But I'll note that the immunogenicity of this vaccine actually goes up well beyond day 28. I showed you data at day 28, day 56, and day 71, and it actually even keeps going up beyond that. But you can see that although it was not the primary endpoint of the study and therefore can't be reported as the primary endpoint, after day 48 in this study, at least with the data we had until February, there is actually no more cases of severe COVID disease in the vaccinated individuals. So after day 48, there was essentially 100% protection against severe disease. And this led to FDA emergency use authorization on February 27th and the subsequent rollout in both South Africa and in the United States. In some unpublished data, we've also looked very closely now at immunogenicity against viral variants to try to explain, because it was somewhat perplexing to all of us, why did this vaccine give robust protection against both the resistant P2 variant in Brazil and the resistant B1351 variant in South Africa, despite what we knew at that time would be a substantial loss of antibody responses. So to try to quantify that, these are data from our phase 1 study in which we set up assays against the original strain, Washington 1 2020, the D614G variant, and then the B117 variant, the California Cal 20C variant, the Brazil P1 variant, and the South Africa B1351 variant. These are neutralizing antibody titers on day 57 and day 71 following vaccination. And you can see that compared with the original strain, there was about a 3-fold reduction of NUC titers against P1 and a 5-fold reduction of NUC titers against B1351, less so for the other ones. For binding antibodies, there was about a 3-fold reduction of binding antibody titers to RBD, which is a little bit less than neutralizing antibody titers. But then together with Galit Alter, we looked at other functional antibodies, other FC functional antibodies, antibody-dependent cellular phagocytosis, neutrophil phagocytosis, complement deposition, and NK cell activation. And for these FC functional antibody responses, there was really a minimal change against the B117 and B1351 variants. Then we looked at T cell responses, which we had hypothesized may be important, particularly in prior non-human primate studies that are published now. I didn't have time to review them today, but we had shown explicitly in non-human primates that CD8 T cell responses contribute to protection against SARS-CoV-2 challenge. So we also looked at T cell responses, and here by ELISpot assays, we actually observed that there was no detectable difference in T cell responses against these different variants, both on day 57 and day 85. And if we look by multi-parameter ICS assays, then there's also no difference in CD8 responses or CD4 responses against these different variants. So basically, these immunogenicity data show that vaccine responses, the neutralizing responses are reduced substantially, 5-fold to B1351 and 3-fold to P1 variants. There's some impact, but less so, on binding and FC functional antibodies. And there's really no impact at all of the variants on CD8 T cell responses. So the robust protection against severe disease with the variants at least raises the possibility, well, it raises 2 possibilities, either that the residual neutralizing antibody responses are sufficient to achieve that degree of protection, or that other immune parameters other than neutralizing responses, such as FC functional antibodies and or CD8 T cell responses, might also contribute to protection. So I'll raise that not as a conclusion, but rather as a possibility. So as probably everyone knows, a week ago, last week, there were reports of thrombosis and severe clotting in 6 or 7 patients out of about 7 million doses given with a J&J vaccine, which led the CDC and FDA to recommend a pause of vaccinations to allow time to review the data and to see how extensive this problem was. And over the last essentially week or 8 to 9 days, then there's been a lot of information about so-called vaccine-induced thrombotic thrombocytopenia. 6 to 8 cases so far, probably a couple more that will be reported tomorrow, but not a deluge more. in about 7 million vaccinations. This is clinically characterized by severe thrombosis, including a predilection for the brain, cerebral sinus venous thrombosis, together with thrombocytopenia, which is a rare combination clinically, because normally thrombocytopenia results in bleeding, not thrombosis. So therefore, it clinically resembles a clinical entity called heparin-induced thrombocytopenia with antibodies induced against platelet factor 4. So the key thing is that this condition is best treated with non-heparin anticoagulants, and the use of heparin in the setting of platelet factor 4 antibody-induced thrombosis actually can dramatically worsen the condition. So that's one of the important reasons for the pause, is to make sure that this syndrome is treated correctly with non-heparin anticoagulants, which will likely be much more, I mean, it's critical to treat with non-heparin anticoagulants. The other interesting feature is that this same syndrome was reported a week or two prior with the AstraZeneca vaccine, which also uses an adenovirus vector. And it appears that this syndrome with the AstraZeneca vaccine is at a substantially higher frequency. Still, the number is sort of to be determined. But it looks like with J&J, it's 1, maybe 2 out of 1 million cases. And with AZ, it's probably maybe a 10-fold higher than that. So this has led over the last week, essentially, to a lot of speculation as to what's causing this. And because these are both adenoviral vector vaccines, then of course, one of the triggers that may be causing this is the ad capsid or DNA itself. Also, a possibility it could be the spike protein. It could be an excipient or contaminant in the vaccine preparations. There's been speculation as to whether there might be very rare inadvertent intravenous administration of the vaccine. We know that IV infusion of ad vectors can cause thrombosis in mice, or something completely different that we're not aware of yet. So the administration of this vaccine was paused as of Tuesday of last week to allow time to assemble the data and to alert the healthcare system. And the CDC ACIP meeting is tomorrow to determine the path forward. So I'll summarize briefly. The efficacy of this vaccine is demonstrated in a global phase 3 study, including in the US, Latin America, and South Africa. There's a high efficacy shown against severe disease in all regions studied and all subgroups, and against resistant variants. We think that a single shot vaccine with no sub-zero freezing cold chain requirements is really quite important for the mass vaccination campaign to reach the hardest to reach people, both in the US and unquestionably globally. And assuming everything can go forward in a safe way, then J&J is committed to supplying the world with at least 1 billion doses of this vaccine this year, and even more than that in 2022. The rollout in the US is currently paused pending CDC and FDA investigation into these rare, very extraordinarily rare but severe cases of thromboses. And this will be reviewed tomorrow. And so as of this time tomorrow, there will probably be a lot more information about this. The pause was implemented globally, and as of a few days ago, the pause was formally lifted in the European Union and in South Africa, and so the vaccine is now continuing the rollout in those regions. And so in the US remains to be determined. So I'll close with a few perspectives. Vaccine development for COVID-19 has proceeded faster than for any pathogen in history. Safety and public trust is critical. Viral variants are going to require further study. Vaccines may need to be updated, and additional variants will likely emerge. And I think we're seeing that with variants originating in many different countries, actually. Neutralizing antibody responses appear to be reduced by the variants, but FC functional antibodies and CD8 T cell responses are largely preserved against the variants. And we feel that multiple vaccines need to be implemented in parallel to accelerate the vaccine rollout in the US and the world. And actually, the variants is one of the strongest reasons to accelerate the vaccine rollout, because the only way to stop the emergence of variants is to control the pandemic with vaccines. So one of the strongest reasons to accelerate the vaccine rollout is to help prevent the emergence of new variants in the future that might be even more concerning than the variants we have today. So I'm often asked, when will the pandemic end? And of course, I can't answer that any better than any of you. But what I can say is that I think that right now we're at a critical juncture. We're essentially in a race between the emergence, between national and, more importantly, global vaccine campaigns and the emergence of variants. And the speed at which we can vaccinate the world and the speed and types of variants that emerge will likely determine how and when the pandemic will end. Will it end more quickly, or will this go on for many years, or perhaps even not end at all? So I'll conclude with acknowledgements about our NHP studies and our clinical studies. And this is a picture of our group at Beth Israel Deaconess Medical Center. This picture was taken, it's way outdated, about 1.5 years ago. So there's a lot of new faces that we have currently in the lab. This is in pre-pandemic times, and hopefully if the epidemic can end quickly, maybe we can get together like this and take more pictures like this again. So I'll stop there and happy to take any questions.
Thank you very much, Dan. As always, very clear presentation, and I was really taken by the data. I don't think I had seen previously that, as you described well, with the vaccine tests that happen to coincide with variants of concern, for example, in South Africa, that those patients still maintain robust T-cell responses. So that's really striking data.
I'll just clarify, the data I showed are actually from a phase 1 study conducted in Boston, in which we tested against those variants. So we actually didn't draw blood in the phase. I mean, we didn't preserve PBMCs in the actual phase 3 trial in South Africa. Now they are. So there will be data from South Africa. But just to clarify, the data I showed you are actually US patients of T-cell responses against B1351.
Okay. Let me ask one more quick question, Ralph. I thought about this, Dan, and I think I probably know the answer, but because, as you said, J&J has used the Ad26 on other backbones, for example, Zika, is there any evidence of thrombosis in those cases? Or is it just that not enough people have been shot up with those agents to actually reach the critical denominator?
There has never been any cases of thrombosis with any other Ad26 vaccines. The largest program, however, has been the Ad26 Ebola vaccine, which has been given to about 200,000 people. So even 200,000 people would be too few to pick up a 1 in a million effect. But it is reassuring that it appears that it's likely not going to be too frequent, because it wasn't picked up in 1 in 200,000. So it looks like it's settling out to be about a 1 in a million. Could go up to maybe 2 in a million, but it's probably not going to go up too much higher than that, at least with the current numbers we have.
Thank you.
And
Ralph, you have a question?
This is a mystery as of now, and is actually triggering almost a new scientific field. It's really quite amazing, because nothing like this, there's no precedent for anything like this before.
So Dan, that was a great talk. And so I have a very general question. So you emphasized the importance of CD8 T-cell epitopes as a potential avenue to provide at least partial protection against variants. And so what are your thoughts about, in future vaccine designs, to focus more efforts to inclusion of more T-cell epitopes? What do you think could be a realistic approach to achieve that?
Well, first, the basis of that statement is a study that we published. I didn't have time to talk about it today, but we published a paper in December in which we showed in monkeys that with adoptive transfer studies, that purified IgG alone can protect if the antibody titers are above a certain level. So we have explicit data showing that antibodies alone can do the job if you're above a certain titer. So the question is, well, what if you're not above that titer? Then, and we did a second experiment in monkeys in which we showed that if you have borderline antibody titers, And if you deplete CD8 cells by intravenous infusion of an anti-CD8 antibody, if you deplete CD8 cells, then it increases breakthrough. So that's pretty good evidence that if you have borderline antibody titers to the incoming virus, whatever that happens to be, then CD8 responses can help restrict virus replication, which makes a lot of sense because that's what they're supposed to do. So that's sort of the basis of the premise, or rather the hypothesis, that neutralizing antibodies alone can probably protect if they're above a certain level, which we don't actually know yet for humans. And there's some conflicting clinical trial data. But that in borderline cases, or if you're below a certain level, then CD8 responses likely can contribute. And whether they contribute to protection against infection or protection against disease or severe disease is sort of a source of debate. And now I've totally forgotten your question.
So my question
vaccine.
is,
inclusion.
yeah, so how do you include, sort of more intentionally, a broader array of CD8 T cell epitopes?
So I think it's important. I think the CD8 responses to vaccines is understudied because of the focus on antibodies. And it turns out that spike is, at least in our studies and those similar data with Alex Sete, that the 3 genes that generate the highest CD8 responses in vivo are spike, nucleocapsid, and membrane. So spike actually is one of the 3 genes that does generate CD8 responses in humans to infection and as well as to vaccination. So one could complement the spike CD8 responses, which you sort of get already because you have spike immunogens. One could think about complementing it with a nucleocapsid or a membrane immunogen, or one could develop conserved T cell regions, either with conserved or mosaic type approaches or epigraft type approaches to enhance CD8 responses. And I think that there's actually some studies that can be, some proof of concept studies that can and probably should be done in animal models to look at that. And meanwhile, I think that the potential role of whether or not CD8 responses are truly important in humans needs to be explored.
Okay, and so then we also have a question from the audience, from Rahul Aurora, and he's asking, what's the primary reason for what he calls the spectacular failure to develop herd immunity against the virus?
Well, I think there's a couple perspectives to that question. So, I mean, currently in the US and nowhere in the world do we have herd immunity yet. And I think, I guess I could answer the question from 2 different perspectives. One is from the perspectives of variants, that it's been clear, at least in select populations, that widespread infection does not necessarily give you protection against a new variant. We've seen that in Manaus in Brazil, where I believe it was estimated that 76% of the population was infected with the original strain. And yet there was a widespread surge with the P1 variant. And we know the P1 variant can partially evade antibodies induced by the original variant. So I think that's one of the clearest examples of how, at least in some settings, so-called widespread immunity from natural infection does not protect against infection with a variant virus. And then the other way to answer the question, I'm not sure if the questioner was asking the variant perspective or the vaccine hesitancy perspective, but is that we're simply not getting vaccines out there enough. Part of it is lack of vaccine supply, which is finally being relieved in the US, still is the major problem throughout the world, that there's simply not enough vaccines. So there's many countries that have not administered a single dose of vaccine yet. And only in select countries has vaccinations been given to a substantial fraction of the population. So the other problem is vaccine supply is not there and won't be there for many, many months to much of the world, as well as vaccine hesitancy, that for many different reasons, a substantial fraction of the population is not yet convinced that getting a vaccine is the right thing for them. So I guess those are the reasons, some biological, some more sociological, and then some just logistic.
Thank you very much, Dan. I think to stay on time, we have to move on. And I promise, Ralph, if you stay on till the end, you'll get the first question in the discussion session, because I apologize for cutting off your question. But we
That's
do
okay.
need to move on to the third speaker. And our final speaker today is Dr. Michael Mina. Dr. Mina is the Associate Medical Director in Clinical Microbiology at Brigham and Women's Hospital and Assistant Professor of Epidemiology at the Harvard School of Public Health. Michael's research combines mathematical and epidemiological models to understand population and immunological consequences underlying infectious diseases, with a specific focus on the development of new technologies that enable high throughput detection of the causative agents. So we are accordingly extremely fortunate to have Michael here today to present his work on COVID-19, strategies for surveillance and control. Michael, take it away.
Well, thanks very much. All right. So this talk was going to be quite a bit different from the last two. It's mostly going to be around how testing for a virus can actually serve to enhance our ability to control this virus in a way that we haven't yet really accounted for during this pandemic, relying almost entirely, of course, on vaccines, which we've just heard quite a lot about. I've sort of made a deal with myself that throughout this pandemic, I would, as much as possible, start with this slide. And this slide isn't so, it's not super relevant to the rest of this talk, but what it is relevant to is that novel doesn't mean unknown. And I think one of the major problems during this pandemic has been that when we applied the word novel to this new coronavirus appropriately, it was taken to assume that we knew almost nothing about it. And we just heard about how much was already known that allowed us to get the vaccines that we have in place today up and running so fast. So we knew in many ways that the virus wasn't, or I should say the vaccine manufacturers and vaccine developers and many immunologists and such knew that this wasn't purely novel from all aspects. But in so many ways, the public health response kind of went back to the drawing board and assumed that we knew nothing. We even saw people saying, you know, we don't need masks. We're not sure if masks will be beneficial early in the pandemic. Obviously, that was for lots of mixed reasons, but it is representative of this idea that we thought we knew nothing as a society and especially in large parts as a public health body. But we know so much, and this is just a slide to show the seasonality of seasonal coronaviruses. We knew seasonal coronaviruses were, of course, seasonal, and that we should have expected when we started closing down in March of 2020. Many of us were saying we need to start thinking about the fall, but many other public health folks around the world were saying, well, we're not sure if this is a seasonal virus. That was just exemplary for me that of course, this is a seasonal virus. We were going to see seasonality, but it had what's called the force of infection. The momentum of the virus across the population was so strong that it was able to continue spreading even in the summer months, for example. And so I hope that in new pandemic times, we can really, we can take a big step back when a novel virus comes around again and rely heavily on what we have already known in the public health sphere, similar to how the vaccine developers have relied so heavily on previously accrued knowledge to get up to speed so quickly, and we don't try to write things over from scratch. Switching to testing. Testing during a pandemic is not medical testing per se. And this, I think, has been one of the greatest confusions around testing during the pandemic globally. Nearly all tests that we usually produce and use in pre-pandemic times are for personal health. They are medical diagnostic tests, and in the United States anyway, they are regulated as such. But during an epidemic like this, the point of testing and all of the drive-throughs and everything that we set up during this pandemic, that wasn't for personal health primarily. It was primarily to know who is transmitting and when. And that's for population health. That's for knowing, am I infectious and a danger to the other people around me? Not so much knowing, do I need to go to the doctor? These are really different things. And by focusing almost exclusively on personal health during this pandemic as the modus operandi for testing, our regulatory framework has been entirely skewed to the point where we actually were neglecting simple fast tests, which I'll talk about, in order to get PCR tests with 10 or 12-day turnaround times, just because they met the metrics for medicine. But these were entirely inappropriate for population health. And that's just to set the stage for much of what this talk is going to be about. But first, I want to talk about briefly, put a plug in for what I think should be done for the future. And there's a lot of different things that we should learn from this pandemic. But one of them is the true need for global surveillance in a way that we haven't yet seen. And so this is a position piece that myself and folks at the NIH, the Wellcome Trust, and at Princeton wrote. And this is based on this idea that the world needs something like a global immunological observatory, a weather system for viruses and for pathogens, a new mode so that we can know where pathogens are at any given time, whether novel or endemic. And we can actually start learning the rules of the game of virus transmission in a way that we don't yet currently understand. Very similar to how the weather system, it wasn't until we set up the weather system, the weather monitoring system, that we really began to understand how to predict and interpret it. The same thing goes for pathogens. And instead of weather buoys in this system, it would be people's specimens. It would be specimens that come from dried blood spots, from discarded samples from blood banks and hospitals and clinics. A consistency across the globe is that people are always taking blood out of themselves or saliva for various reasons. And most of that blood ultimately gets discarded, usually as discarded specimens after pathology use. We could capture those and start processing them in using high throughput immunological tools to actually start writing the rules of how viruses transmit in a way that we don't currently have. We have a pilot study going on with a major company, a plasma donation company right now called Octapharma, to do just this. For example, in the US, we get tens of thousands of specimens into my laboratory weekly, and we're processing them for COVID antibodies. But it doesn't need to just be antibodies against SARS-CoV-2. There's new tools, and this is a technology initially developed by Steve Elledge at Harvard, and that I've been working on quite a bit since then. This is just one example of a high throughput immunological tool called VirScan. And what you're seeing is in the columns of this matrix are individuals, one person, one drop of blood per person. And what you're seeing in the rows are epitopes or antibody responses against those epitopes for different pathogens. And I'm showing 4 pathogens here, but actually the data set is around 400 pathogens. And from less than a single drop of blood now, we can evaluate tens of thousands or hundreds of thousands of potential antibodies inside of a person and profile them. And we can do this for dollars, not for tens of dollars, but truly for single-digit dollars. For almost the price of a simple ELISA, we can get hundreds of thousands of antibody data points now. And so these types of technologies are now making it possible to make something like a weather system for viruses, where with every specimen we monitor the immunological landscape or the immunological profile for hundreds of distinct pathogens. And we can even use these profiles to detect novel pathogens, pathogens that look something like a coronavirus, but maybe have a slightly skewed response to versus what we would have expected for a coronavirus. That we could look at the negative impression, for example, of where antibodies are lacking to recognize that new pathogens are actually circulating amongst us. So there's a lot of different uses for the immune profile that we haven't yet really put to use from a public health perspective beyond just simple seroprevalence and serosurveillance studies. And I hope that in the future we actually learn and we start building these types of global systems to advance our ability to actually contain and control and ideally prevent future pandemics like this. I'm going to switch now. That's really all I'm going to talk about with some of the immunological work. And I'm really going to focus now on viral testing. And this distinction between personal and population health that I think has been at the core of the testing woes that we've had and has been front page news, throughout this pandemic at different times in terms of the confusion by our own regulators, our own CDC and others, and scientists and physicians across the globe about what is a public health versus a personal health test. So for population screening, we need distinct characteristics relative to medical testing. The usual priority order for a medical test, especially for a test like a virus, is usually we want something, if it's PCR, it's giving us extremely high sensitivity. And that's usually priority number one. Of course, specificity is also massively high up on the list. The cost and frequency with which you can give a test is usually not even considered. The speed to get results also are not usually considered when we're thinking about medical tests. But for population screening, the sensitivity issue might not be as crucial as the accessibility of a test. Tests need to be fast and frequent. They need to be convenient and accessible if we actually want to use them to help mitigate spread. And so what that means is that maybe sensitivity has to take a back seat compared to accessibility. The best test in the world never used is not going to be one that can control spread of a virus. The best test in the world, if giving results back 10 days later after the transmission window has closed, is not going to be useful to help control spread. It could still be useful from a medical position, but not for public health. And so for those who aren't familiar, this is one of the real-time PCR instruments that the CDC assay was first posited to use, the ABI 7500. The only reason I show this is where we were at the beginning of this pandemic. We had a CDC and the CDC assay was suggesting to use this instrument with the 3 nucleocapsid primers. The thing that was really lacking though is that to use this instrument requires a lot of expertise. It needs skilled technicians. It's cumbersome. It's not particularly high throughput unless you put it up on robots, which need a whole additional level of expertise. Yet this is where we were at the beginning of this pandemic. This was the standard assay to run to help stop a pandemic from spreading in the US. Of course, I won't go and rehash what happened last February, but there were massive blunders at the CDC. We didn't have any testing in this country until the virus was already largely out of control. Of course, as things moved on into the spring of 2020, we started to see the normal clinical laboratory instruments that we see in any modern day hospital, for example, started to get these tests up and running. Most of these didn't come until spring though. And in fact, when I spoke with folks at Roche and Hologic, for example, early in the pandemic, and I said, are you guys planning to build a COVID test? In late February, there were still questions about whether they were really going to bother building a COVID test, which was astounding to me. And it just shows that we didn't maybe have the right public health messaging at that point. But we do see new tests, but all of these tests are generally performed in a lab or are going to be limited by either their scalability on the top. They're all cartridge and fast and easy, but they're not particularly scalable. Or on the bottom, they need to be in a laboratory setting. Once the sample gets into the lab, it can be fairly quick, but it does take a long time to line them up and queue them up. And sometimes that was days or even weeks during this pandemic to get the specimens through the laboratory. But then, and now we're seeing a massive shift. We're seeing tests like these rapid antigen tests, rapid paper strip tests, and these have been a topic of extreme debate during this pandemic. These tests are antigen tests. They don't have an amplification step. There's no PCR amplification that gets performed at the beginning, although some of those are now coming along. But the vast majority of these are simple tests that work very much like a pregnancy test. If you have 2 lines, you're positive. If you have 1 line, you're negative. But instead of sending them off to a laboratory somewhere, they can be done at your kitchen table or in your bathroom at home. And so these could potentially, if they work, could be massively important to stop spread without incurring at the peak of this 10 or 14-day delays. And but the question has been, are they sensitive enough? Are they good enough to actually be used in a pandemic? And I think that the question has been off base. If our question is a public health one, what tests do we need to prevent transmission? These tests, which I'll show over the next few slides, should be are actually much more effective than a laboratory-based test that requires a send out. And I'll describe why that might be. The first thing to know is to understand a bit about viral replication inside the host. This is just a schematic. It's generally pretty reflective of what happens in the average person. When you get infected, there's a period of time during the incubation period when no test is going to find you positive, and you're going to be below the limit of detection after exposure for a number of days before even the PCR test can detect you as positive. And but eventually, for many PCR tests, the limit of detection might be 1,000 viral copies or RNA copies per mil. And so that would be a good test. And so when you ask, well, what's the limit of detection for a rapid antigen test, one of these paper strip tests, it might be 100,000 or 1 million copies per mil. And so that sounds like a massive difference. 1,000 is much better than 1 million. But from a public health perspective, the viral load itself is not the only thing we need to be paying attention to. In this context, we need to pay attention to what is the actual meaning of that difference. If it's a 1,000 versus 100,000 or 1 million, what does it mean from a public health perspective? And that's where time needs to be taken into account. And here what I'm showing is that the time between going from 1,000 copies per mil to 100,000 or 1 million is minimal. It's usually less than a day, in fact. From the moment you turn positive on a PCR test, most people by the end of that day will have turned positive even on a lower sensitivity antigen test. And that's because the viruses, all they know how to do is grow exponentially in us. They don't know how to do anything else. And so when they grow, they either grow exponentially or they get cleared exponentially. And so by the time you're PCR positive, you have enough virus in you that the virus is screaming north. And within 8, 12, 15 hours, maybe 24 hours, you will largely have passed the threshold for both tests. And so what you're losing out is really just 15 or 20 hours of detection. But what you're potentially gaining from a paper strip test is days earlier detection because you're not sending the test out, or accessibility and the frequent use of an inexpensive test. And what's important also on this slide is that almost all of the virus that somebody has is contained just in the peak of your titers. 99% of, on this slide here, I'm showing live virus on the blue, and the RNA is schematically drawn on the dotted line. But 99% of all the virus is really going to occur in this one short little window of time, which is maybe something between 4 and 8 days duration in most people. And that's where the contagiousness happens. That's where you're transmissible. So all of the viral load is really captured there, but then people will stay PCR positive for weeks. And what we've seen is that when comparing to PCR, antigen tests can oftentimes look very insensitive. But the point is that it's not, the insensitivity isn't when you're infectious, and it's not even really at the beginning phase, except for those 15 hours. It's the potentially weeks after being contagious that you remain PCR positive, because the RNA is remaining and still serving to get cleared. And the PCR is so sensitive to detect even a single copy potentially, that PCR will remain positive. Meanwhile, a lower sensitivity antigen test that looks for the whole virion is going to turn negative. In general, by day 10, you have essentially no sensitivity to detect any virus using an antigen test, even though most people will still remain positive by day 10. And most of the epidemiological data shows that by day 10, people are no longer transmissible, which is why the isolation period is 10 days as a conservative metric. So what this is really suggesting is that PCR is the tool to combat this virus. While very specific for RNA, it's poorly specific for people who need to isolate. And this one feature has been extraordinarily confusing for many throughout this pandemic. Just to show that same slide But to put it in perspective, we usually show CT values like I was just showing. And this is just another schematic of those same CT values. But what's important to know is CT values are on a viral load. And this is to just point out that what I was saying about the 99% of all the virus being in that one short window of time. If we convert this logarithmic curve showing CT value distributions of the viral load over time into a linear curve, these two curves are identical. On the bottom is the linear scale, on the top is the logarithmic. And this is just to point out that you have to zoom in to the bottom graph, the linear curve. You have to zoom in 1,000,000 times just to see that little section between CT values of 35 and 40. But it's that CT value, the 35 to 40 or the 30 to 40, that has largely prevented many much more accessible tests from coming to market, because we've been solely focused on capturing virus up to a CT value of 38 or 39 or so. Meanwhile, the transmission is complete by that time. And almost all, we have foregone tests that could detect people at the peak of their transmissibility, just so that we could still capture them at, say, day 15 or 20, when they no longer need to be identified as positive from a public health perspective. So for medical diagnoses, our focus has been on sensitivity and specificity, things like positive and negative predictive value. These are the key measures of test effectiveness. But these are concerned with the performance of the test when you assume that a specimen is able to be in hand. And the accessibility and a medically relevant turnaround time are just presumed in the hospital, for example. You assume that you only need to test the samples that you actually have. But from a public health perspective, you also have to be considering, how do you get more tests performed? How do you actually capture more specimens? And how do you give turnaround times that are relevant to transmission? And this is, if there is no access to the test or limited, or if the speed of the test is very slow, then essentially even the best PCR test and the best diagnostic test in the world will lack any public health effectiveness, which is exactly what we saw at the peak of this pandemic when we needed tests the most. Nearly 100% of tests were being returned in a window of time that was entirely inconsequential, or was entirely abrogating any of the utility of the test to prevent transmission. So this is just a thought process, a quiz for you all to do. If you're charged with limiting spread in a school or a business or in the public at large, you can either choose a 100% sensitive lab-based test with a 1 to 2 day turnaround time, or a 70% or 80% sensitive rapid test that gives you results in a 15-minute turnaround time. Which is the more appropriate test to choose? And this is just an example to help answer that question. In this case, we have this talks about why speed is so much more crucial than sheer sensitivity. If you have a PCR test or a rapid antigen test, and you have 5 infected people in the black here walk into a school or into a building, and all 5 of them get a PCR test, a swab, that's going to be returned in 2 days with a result. All 5 of those people will get detected. And so it's 100% sensitive, totally terrific test. But from an epidemiological perspective, you have 10 person days of walking around potentially infecting other people. So that's just putting it in simple terms. Those 5 people over 2 days is 10 person days walking around waiting for the results, but not necessarily quarantining while waiting, because we obviously are not having everyone quarantine while waiting for results. On the other hand, you have a rapid test that has, in this case, 80% sensitivity. Those identical 5 people walk into that same building. 4 of them are able to be, if screened at the door, 4 of them are able to be identified immediately. And 1 person gets through. Ideally, that 1 person also has a lower viral load, which is why they're not being detected initially. But so that 1 person then walks around for 2 days before getting another rapid test. In this case, you have 2 person days walking around infectious. So in this case, an 80% sensitive test is much, much more effective from a public health perspective than 100% sensitive test. Yet we have focused only on sensitivity as the primary metric at the FDA and in public health and regulatory agencies across the world during this pandemic, because it's the medical metric. But speed is unfortunately not even included in the FDA's thinking about what test to authorize, even though speed is probably the most important piece if we're trying to stop transmission. So a 70 to 80% sensitive rapid test is going to be much more important if your goal is to stop transmission. Another way to think about this is that the primary window of transmissibility of this virus is about 6 days. If a lab-based test is returned after 48 hours, for example, what's happening here in Massachusetts with our schools, we're doing pooled testing with a 48-hour turnaround time, and that's still generally average across the United States. Then what this means is that for every case, we lose a third of the test's effectiveness to stop transmission. And for every 3 cases that we detect, even if we detect them at the beginning of their viral load, you miss a full transmission window. So even if it's a 100% sensitive test, if it has a 48-hour delay, and we assume that the average duration of real transmissibility is about 6 days, then that perfect test can only max out at a 67% effectiveness to curb transmission. And so that's really the baseline that we should be working with when we're considering the PCR as a gold standard, not to mention that it's not specific to the transmission window. So it will cause many, many people to have to isolate who no longer need to be isolated. And frequency matters. If you're not testing frequently for a virus like this, because of asymptomatic transmission and because of pre-symptomatic transmission, which is extremely common, you're largely going to miss the transmission window either way. And so if you're testing daily or daily would be extreme, but let's say twice a week, you can actually have massive impact to reduce spread of this virus. And that's because the virus has had an R0 of around 1.3. So all you have to do if you have an R0 of around 1.3, you just have to figure out a program that's going to allow you to get R below 1. And I'll show that on another slide right after this. But the point of this slide is to show that it is not the high versus low sensitivity. And this is based on a lot of mathematical modeling and agent-based models that we've built that include viral kinetics inside of these little synthetic agents that walk around and bump into each other. Essentially what this shows is that if you're testing twice a week, it doesn't matter which test you're using, high versus low sensitivity, because you're not going to, because all of the tests will detect you when you're transmitting. So as long as it's frequent, you're very likely to catch people at the beginning of their infection before they go on to infect on average 1 or 2 or 3 additional people. On the other hand, if you start to get into weekly or every 2-week testing of your community, then you're really going to start losing your ability to actually stop infections with a test program. And to put this in context, this is assuming a whole community is getting tested every 2 weeks. Most people in the United States have never been tested. Only about a third of the United States has ever gotten a COVID test during this pandemic, which is a testament to how inaccessible testing has largely been for most of the country. And of course, as I was just mentioning, it's not just the frequency, it's also the turnaround time. Even with a frequent testing program, if turnaround time becomes 48 hours, you start to lose most of the benefit. And if the turnaround, and this, when we did this, we hadn't ever even considered the idea that turnaround times might turn out to be 5 or 7 days as they were at the peak of this pandemic, at which point they were effectively useless. So we spent millions and millions and millions of dollars, billions of dollars to get all of the drive-through testing sites up and running, essentially for a useless test across the country, which of course was reflected in out-of-control cases. And this is to give an example of what happens with pandemics or with viruses like this. Viruses are simple, as I mentioned earlier. They either go up exponentially or they crash exponentially if left to their own devices. And in this pandemic, we have had a virus which, along with other control measures, has generally had an R0 of around 1.3 or an effective R of around 1.3. What that means is that if you start with 100 infections, then over about 30 to 35 weeks, those 100 people on that given day are going to turn into 600 new infections per day. So that's because exponential growth. R of 1.3 doesn't sound like a lot, but over a month, it becomes very high. On the other hand, if you were just able to get those 100 people not to infect 130, but just to infect 90, you just need 100 people to go on and infect 90 or even 99. Then after that same period of time, after 30 to 35 days, instead of 600 new infections per day, we would have had 40. So the test program, if we were to put something in place, doesn't have to be perfect. We don't have to stop everyone from transmitting. We just have to stop 100 people from infecting more than 100 people, which is actually quite doable with frequent, scalable, accessible tests sent out to the whole community. But we have instead focused so much on making sure that every test we used was perfectly sensitive, even at the expense of utility, which was a massive problem. And this is from that same agent-based modeling framework that we build. We can ask the question, how quickly would a test program, if scaled up to the community level, actually work? And in this case, what we can see is if we had an outbreak that was raging upwards at a prevalence of 4%, if we started a program of frequent testing back in August or September or October, then even if half of the community completely disregarded their results, 10% of the tests completely failed, it still would have been enough testing to get R below 1 and bring that outbreak under control. And had we had these tests at the end of last summer and early fall, and had we authorized them in time for frequent use by the public, we could have potentially prevented the surges of the fall and winter and potentially prevented hundreds of thousands of additional deaths by keeping R below 1. And so for public health, a lab-based PCR test is very sensitive. But really for public health, we need tests that are accessible, that can be used frequently, that can be fast, give actionable results, that can be that have very low capital costs. You can do them at home, that can allow equitable testing across the community, and ideally tests that are mostly specific for the transmissible period, so that we're not erroneously isolating millions of people who no longer need to be isolated. Unfortunately, the lab-based PCR test generally fits one of those categories, but really doesn't fit most of the categories that one might hope for with public health. And just to be clear, I'm not against PCR. It's what I normally do for my medical position, and I helped start the Broad Institute's PCR program. But I recognized very early through that effort that it wasn't going to be the right type of test for really working on public health, unless you could do it as we did with the schools, which was twice weekly testing with a PCR with a very fast turnaround. And that's only because the Broad Institute is so good at what it does, which is efficiently testing. But that's not easily replicable across the country. So while we have focused so heavily on the sensitivity of tests, and all of our regulatory landscape has considered sensitivity as core and crucial to allowing a test to be authorized, we have done that at the expense of making a test that can actually be useful for public health in this pandemic. And I would posit that the optimal priority order for a public health test should first and foremost be frequency and accessibility and speed. And the sensitivity, because again, the virus transmits not at 1,000 viral particles per mil, but at 1 billion or 1 trillion viral particles per mil, the actual sensitivity, if the test is fast, doesn't need to be high. It can actually be very moderate at best, and still be a much, much more important tool for public health. So why do studies show such low sensitivity? I've already touched on this, of rapid antigen tests among asymptomatics. And we wrote a bit about it in a piece in The Lancet recently with some folks from the UK. But I want to show a simple model to explain why so many studies, including from our own CDC, show 30 to 50% sensitivity of the rapid antigen test amongst asymptomatics, and why that is okay. In fact, it probably represents a very, very good public health test. And so we can just take a very simple model on the left here. And the simple model is just to ask, this is a basic SIR compartment epidemiological model. And what it does is we have people who are infectious for 10 days. They are then post-infectious, but PCR positive for another 10 days, which is generally what we see in this pandemic. Actually, it's probably more like infectious for 7 or 8 days and post-infectious for potentially 15, 20, or 30 days. And what you can see on the bottom is what we would expect amongst asymptomatic people if you have a test that is perfectly sensitive and specific for the period of time when people are supposed to be isolating. So if you're supposed to be isolating for 10 days, and then you remain PCR positive for another 10 days, then the expected sensitivity of the rapid test that we would observe compared to a PCR test would only max out here in the dark black line on the bottom chart. At the beginning of the epidemic, it would look like a 60 or so % sensitivity against PCR. But then towards the end of the pandemic, or on the end of an outbreak, and I won't go into detail why it changes, but it does, you should only expect a maximum sensitivity of a rapid test relative to PCR of less than 40%. And that's because the PCR, it's not because the rapid test is missing something crucial for public health. It's actually because the gold standard is the wrong gold standard if our interest is transmissibility and who needs to isolate. PCR simply isn't specific, which means that if we actually follow people and question how sensitive is a rapid test for a public health test against a medical test, it's going to naturally look ineffective when actually that's an illusion. And it's driven by a gold standard that's not specific for the public health question at hand, which is, do I need to isolate? And in fact, what we saw in Massachusetts in June and July was that the vast majority of people who were positive on our surveillance programs and were asymptomatic in June and July in Massachusetts had cycle threshold values that were far below, or I should say far above, viral loads that were far below what would be deemed possibly realistically transmissible. And this is just showing them on the bottom left here. This is a graph. Each of those violin plots is made up of 1 week of specimens from Massachusetts in June and July. And so that's 8 weeks worth of specimens. And what you can see is the median cycle threshold value in Massachusetts at a period of time when the outbreak was at a lull was around 35, if not even higher. So my best estimate is not a single one of those people was still transmissible. And probably not a single one of them actually needed to isolate, nor be contact traced for their contacts over the last 48 hours. If contact tracers were going to go in and contact trace them, they would have had to ask, who were you having dinner with 2 weeks ago, not yesterday and the day before? So by focusing so heavily on an extraordinarily sensitive PCR test for asymptomatic surveillance of people, we not only isolated people after their isolation window and their transmissible window had already closed, but we also contact traced and quarantined all the wrong people. And it's why when the fall came around, we had almost no ability to actually use testing to help slow transmission. And on the top is just a piece we published in the New England Journal about this, which discusses the frequency and why frequency of testing and sensitivity of a test in the context of public health should really be asking, what is the sensitivity of a testing program to detect infectious people, more than what is the sensitivity of any individual test to analytically evaluate whether a virus exists. And if you're not testing frequently with a sensitive, but you're using a very highly sensitive test, then it's much more likely that you're going to find people who are PCR positive, but post-infectious. And in fact, just today, I believe in JAMA, there is a paper showing this, which using the NBA, Jonathan Grad here at Harvard was part of the author list, was essentially looking at NBA players who were tested and who continued to be testing positive long after they were infectious, or sorry, long after they were first identified. And there was no evidence of any transmission after people had already recovered symptomatically, but yet remained PCR positive. So rapid tests can help to reopen the economy and keep it open. And this is just an example of two big studies that are happening now. One is a study that I'm the PI on, and this is working with Citibank to use tests to help them evaluate, to essentially evaluate how at-home testing can be used to prevent transmission in the workplace. And so in that study, individuals are using a test Monday, Wednesday, and Friday at home before going to work with the hopes of identifying themselves as infectious beforehand, before they actually go to work and cause many other people to either become infected or needing quarantine. And then the CDC and NIH recently launched a massive effort using rapid tests. Just a few weeks ago announced this. In Tennessee and North Carolina, they're giving 2 cities as many tests as they want, up to 3 a week per person for free, to see does it help diminish transmission at the community level. And this is something that Francis Collins and Bruce Stromberg have really pioneered that just started. And so we'll be seeing results of that very soon. And so I just have 2 more slides, and this is to explain an important feature about testing with an antigen versus a molecular test, because we're going to start seeing more and more rapid molecular tests as well as rapid antigen. I think of a rapid antigen test as a red light test. This is a test that if you're negative, you keep doing everything else the same. But if you're positive, then you isolate and you quarantine. And by the same, sorry, if you're negative, I mean, if you're still in a period of time when you're wearing masks, keep wearing a mask. If you're still distancing, distance. Of course, this is shifting radically now with vaccines. But if you're positive, isolate and assume you're positive. These new molecular tests, however, which are now becoming over the counter even, are going to be considered green light tests. If you're truly negative on one of these, then at the very least for that day, you can assume that there's very little risk of you transmitting at all. And you can go about your day, probably go to work, not bother wearing a mask in a board meeting, whatever it might be. And so I think of green light tests as molecular tests. So will testing be needed in the context of vaccines? Of course, this made the news yesterday or 2 days ago, whenever it was, of an unvaccinated worker set off an outbreak. This is probably making a lot of people nervous to see these headlines, but we also have to take the headlines seriously. And in particular, my concern is that with variants and with elderly people, and in particular, being the vulnerable people we're trying to protect during this pandemic, there are decades of research showing that elderly people and vulnerable people in this pandemic just do not hold on to immunological memories, if you will, and protection as well as younger individuals. And so one concern is that once we get to this fall, we may see small surges that are generally driven by younger individuals. But if those surges of cases bleed into the older populations like this example, we might end up seeing by the time fall and winter come around, significant waning of immune protection in the most elderly and thus most vulnerable. When we couple that to slight diminishments in immune protection amongst variants, we may still see surges of cases happening, particularly in places like nursing homes and senior centers. And I would just say that testing is our eyes during a pandemic. If we don't have tests, we don't know where viruses are. So to think we don't need testing anymore is a little bit short-sighted from a public health perspective. I think it will be crucial, just not nearly in the US anyway, the extent to which I was just talking about needing to use these tests to really stop transmission at a massive public health level. So to conclude, I want to say that public health is just not the same as medicine and shouldn't, and I think we recognize that, but it shouldn't be selectively considered as such, especially in our regulatory agencies. For the next pandemic, we absolutely need new measures of sensitivity. We need new ways to evaluate things like tests. We need new ways to even do vaccine trials that really account for public health and accessibility, which led to all of these discussions around vaccine dosing, for example. We need to consider public health metrics distinctly from individual health during a pandemic like this. And in the US, what I would say is despite using these very, very sensitive PCR tests, unfortunately, our sensitivity from a public health perspective to actually detect cases in time to act has generally been around 5%. And that is extraordinarily low. And it is, I hope, will help us think differently in the future about how to evaluate things with a public health mindset. So some of the folks here that really worked on this, James Hay in my laboratory, Dan Larimore is at Colorado, and his lab is in Colorado, Brian Wilder and Milan Tombe at Harvard, and some other folks who have been crucial to these discussions.
Thank you very much, Dr. Minna. That was an excellent talk. As a virologist, I was really taken by the discussion of R0. I actually am one of the people on the call who understand that. And I think you really elegantly described that about how rapid testing with less sensitivity can get your number down. And that's really crucial in these types of pandemics. We went a little over. So do any of the panel members have questions for Dr. Minna? Not, I think we can just segue to the final. I think we have about 8 or 9 minutes left to the meeting, and then that was dedicated to open discussion. Dr. Barrett, do you have a question?
Yeah, Michael, that was a great talk. I really enjoyed it, and I really enjoyed it from the public health perspective. Clearly not taken into account for testing during the pandemic. Your transmission data arguing that a certain CT value is critical for transmission. How tight is that prediction? Is that a gut feeling, or is there solid epidemiologic data to support a rate of transmission based on CT levels?
Well, I would say that the real critical piece there is the time window, is the epidemiology that we have. It's very hard to actually get somebody's CT value when they're transmitting. In fact, every time it's been done, it ends up leading to massive biases in the data, because people have tried to say what was the index case's CT value, but they're actually measuring the CT value usually 5 or 6 days later, or 2 days later. We do have very good data, however, that shows that, well, I think I can show one slide here to show you that the correlation between this one here. So this is data of CT values over time in people who were being monitored pretty much daily during quarantine. And what you can see here on the top right graph, those are the CT values of individuals followed over time. And most of them had their most recent negative PCR result a day earlier, or maybe 2 days earlier. What you can see is almost everyone on these antigen tests turns positive during days 2 through 6 or so, 2 through 7, who is at a CT value that is both low and is within this window of time of transmissibility. And we have very good epidemiological data, better than the CT value data, to show that it's really this first period of time when people transmit. And they generally don't transmit after day 7 or 8 or so. So while the CT value itself might not be the best metric, because there's so much individual variation, what we do see is, even without considering CT values whatsoever, the antigen tests appear to pick up pretty much everyone. That's the blue dots here on the top right. They appear to detect everyone who needs to be detected during that period of time. And then once you get to day 8, 9, or 10, you see that PCR, many people, I'm not showing it here, many people then stayed positive for 20 or 25 days. And in general, the antigen test turned entirely negative by day 10.
All right, thanks.
Yeah, you made the point really well that especially at the tail phase of the infection, after you're post-transmissible, that PCR is basically wasting resources, right?
It is. I think, one of the most frustrating things for me has been this, there has been an intense debate, especially in the UK, around false positives with antigen tests, for example. But in my opinion, isolating somebody who no longer needs to be isolated, who's at day 15 of their infection, isolating those people from day 15 to 25 is as destructive as a true false positive altogether. And I do think that that needs to be taken into account when we think of false positives. It shouldn't just be analytical false positives for public health. It should be relevant false positives. One of the reasons this has been so hard and such a long slog to make these points over the years, because we literally don't have a science or definitions or a language around these ideas. From a regulatory landscape and many other landscapes, we haven't had the right words to even use. And so much of this year has been spent trying to develop a vocabulary around how to talk about a public health test versus a medical test.
It's a very complex problem. I think you alluded to it towards the end, even if you could roll this out, and I'm glad to see that there are targeted rollouts now. You need compliance. I mean, one thing I think a lot of us face and think about a lot is the quote general science ignorance of the general public. So you got to have people use it, and I think your modeling took into 25% misuse, for example, in some of your calculations, but it's quite a complicated public health issue as well for those reasons.
It absolutely is. Yeah.
So, Dr. Min, I have a question for you. Since you're so involved in the testing world, particularly with the broad, what is your current assessment of the effectiveness of masks? There's been a lot of debate on this topic. There have been studies here, for example, at the Brigham, but those were months ago. So, what's the current data on that topic?
I think from sticking with the scientific, or at least the general flow of scientists, masks generally work, but exactly how, what role they really have in quantifying that, I think is very, very difficult. For example, if you're sitting in a single place for hours at work, for example, and you have a surgical mask on, you're circulating air throughout your mask and into the environment. It's not an N95 type of mask, which really has any sense of a seal. And so when you start to get into that type of thinking, it does, I would say that it raises the questions of just how effective is a surgical mask in that type of environment. My personal stance is the data isn't great in either direction, and I'd rather be safe than sorry with a pandemic like this, and have people wear masks, with reason. Obviously, don't wear masks if you're in your own house, but when you're talking very closely with somebody, I would say it's probably better than not wearing a mask.
Have you done a lot of modeling, Dr. Mina, about the way that the rapid test will affect, for example, the rate of what are not values will change as the vaccine is being rolled out? I mean, that's another highly complicated variable to take into place, but it's really at least where the US seems to be moving towards.
Yeah, one of the things that I think will be the place for rapid tests in the U.S. moving forward, thanks to vaccines, is we won't need that same incredible level of. We're no longer trying to mitigate all transmission with a test. We have another tool, but I do think that we will continue to see outbreaks happen. They won't be huge, but they will happen, and they will happen more frequently as the winter sets in. And what I would like to see is that we have another tool at our disposal that allows a school not to have to close down for 2 weeks because of it. And the nice thing about rapid tests, more than other types of testing programs, is they can be dynamic. You can have a test that you say you totally turn off. Nobody's using the test. And at the flip of a text message, you can ask parents to start testing their kids again, as long as they already have the test in their home. So tests can be really dynamic. If you have a big outbreak, like the one that was in the New York Times, what they did was they actually scaled up rapid testing very quickly, used them daily, in fact, for everyone for a week, and then quickly scaled back once the outbreak was under control. That is one thing, we continue to live in this world where we think this is our testing program and we've got to stick to it. But these simple tests, because they're so simple, so accessible and easy to move around, they really can create a new dynamic type of testing program. But the first thing we have to do to make it work is get them into the hands of people so that it can actually, so that they know what they are, they're familiar with them, And one thing I've been saying this whole pandemic is, for whether it's for vaccines or tests, is that we should be getting some of the best media agencies involved to help the public understand what is a vaccine, how effective are vaccines, how effective are tests. We should have, for those people who remember all the Coca-Cola ads with polar bears on skis and such from the Olympics, those are very effective messaging. We should get those same companies to get public health messages out to the country to help people understand these things.
Yeah, I think we basically have to close the meeting now, but I think you've made it pretty clear, and I think it will really help depending on how effective the vaccine is, and to inform all of us how frequently we might need vaccination. If we could have these types of tests embedded, it's almost like real-time monitoring of viral blips, right? It's going to help you with that as much as anything. So, hopefully Dan's work and everybody else's work around the country to make really effective vaccines will in large part mitigate this. But I think probably we all expect this virus is going to be here, right? It's going to remain a seasonal virus, and we'll have to see what the future holds in terms of severity to public health and also frequency of vaccination. Kai, do you have any other closing remarks?
Just thank you to all of the speakers. That was great.
Yeah, thanks a lot, guys, for taking time. We know how busy you are, and it was really a fascinating meeting with different perspectives on the current pandemic.
Thanks a lot.
Thank you all.
Thank you very much.
Take care.